Welcome to this lecture on histology. This will look at microscope slides of nervous tissue, the motor neuron specifically, and all three of the muscle tissue, skeletal, cardiac, and smooth. The first slide is of motor neurons. We can see that being listed here. And it tells you the objective magnification here. This is not the total magnification. If you want the total magnification, you have to take that times 10x. As we can see, these are motor neurons, as indicated by the arrows. We can see them throughout the picture. We can also see representative examples of dendrites and axons. Dendrites are what is receiving the signal towards the neurosoma, and the axons are what is passing the signal away from the neurosoma. Here's a closer view. This closer view is at 10x. That is for the objective. Here are our motor neurons again, with our representative dendrites and axons. One more slide showing an extreme close-up. We can see the neuroglial cells. Those are the cells that are aiding the motor neurons in their day-to-day activities. Here is our axon, our dendrites, and this structure right here is the nucleus. This is a 40x objective. Here is our first muscle slide. This is of skeletal muscle as indicated here, and this is a 40x objective. A couple things, we can see the striations. Those are the bars that you see going across the muscle fibers themselves, and these purple dots are the nuclei of those muscle fibers or cells. You can see those located throughout. Now keep in mind that skeletal muscle is not branching. These are actually tore here, that's why they appear that way, but they run in parallel fibers and unbranching. They are voluntary, and they do not have any gap junctions. Take and compare skeletal muscle to cardiac muscle. The two can be easily confused because you see striations within cardiac muscle as well. However, the way to tell the difference is the more prominent nuclei that you see here. There are branching muscle fibers and the intercalated discs, which act as gap junctions that allow the signal to pass from one cell to the next. Finally, we have the smooth muscle. This is a 10x magnification, objective magnification. First thing to note that there is no striations. Each of the purple dots are nuclei that you see throughout this slide, and that they are spindle shaped. They're going to look something like that. For a closer look, we can see again the spindle shaped nuclei. This is at a 40x objective, and some instances of muscle fibers within that tissue.